My name is Dr. Chris Jenkins, and I am the CEO of the Orianne Society and the host of the Snake Talk podcast, the podcast where you learn about nature's most feared, maligned, and persecuted animals. I invite you to listen to this conversation, and maybe you'll find that what you perceive as fear is actually rooted in a deep fascination. Welcome to the Snake Talk podcast. I am sitting here with Dr. Stephen Mackessy from the University of Northern Colorado, where he is a professor, and we are going to have an interesting conversation today. He is an expert on everything venom, and in particular has done a lot of work around kind of the biochemistry of venom and, for example, how it could be utilized in the medical field or for other things. He's done a lot of ecological and natural history related work. So I think we're going to have a great conversation. And for those of you that listen to every episode, you will remember Steve has actually been on the podcast before. He was part of the panel episode that we did at the Northwest Partners in Amphibian Reptile Conservation meeting. So welcome back to the podcast, Steve. How are you doing today?
I'm doing pretty well, Chris. Thank you for the invitation to talk about one of my favorite subjects, venoms of snakes.
Yeah, great. Well, you've been on the podcast, and we probably talked a little bit about your background, but I want to dive into it a little bit deeper. So I want to start off at the very beginning and figure out how was it that you would get into something so strange as snakes and venom and these types of things? Is this something that bit you, pun intended, I guess, as a child, or something that hit you later in life?
Oh, it developed pretty early on, and it seemed kind of a natural move for me. I grew up in Southern California, born in San Francisco, but moved to Southern California in a town called Monrovia, which is right against the San Gabriel Mountains. And so when we were kids, we'd go hiking all the time and occasionally see snakes. So I was interested in snakes then. As I started in my early teens, a friend introduced me to another person who had just opened a business. And this was a place called Western Zoological Supply in the same town that I grew up in. And it turned out, it developed that I worked there as a job all through high school. And I was taking care of venomous snakes from all over the world. So everything you can possibly imagine, everything you've ever seen in zoos, we had there, because they were a supply company that brought things in from overseas and then supplied zoos and schools with all sorts of different venomous animals. And that included things like mambas, king cobras, spitting cobras, vipers of every flavor and type you can imagine from around the world. So that got me quite interested in diversity of snakes. Early on in that, as a 15-year-old, I had a little accident of sorts with a Southern Pacific rattlesnake where I picked it up with a snake hook like you're supposed to do. And then I decided to hold it by the tail like you're not supposed to do. And it very quickly turned around and hit me in the left hand. Long story short, it put me in the hospital for about 6 days, swelled my arm up about double into the shoulder, and gave me quite an incident to talk about. So,
if I remember right,
so those were kind of early events.
Southern Pacific is actually fairly toxic as far as North American rattlesnakes go.
Yeah,
is that true?
It's a reasonably dangerous kind of middle of the road rattlesnake, tending towards the more toxic. Well, as it turns out, if you're going to be bitten by a rattlesnake, it's much better to be bitten by a baby rattlesnake, because contrary to popular opinion, they can actually control the venom that they're injecting. Their venom is a little bit more toxic than the adults, and what I found later as part of my master's work is about 2 times as toxic drop for drop. But the amount that they can give you is so much smaller than an adult snake. And like any compound or drug, dose determines effect. And so a small snake can only produce about 10 microliters of venom, whereas an adult Southern Pacific rattlesnake can produce 500 microliters. So the difference there is dose dependent.
Yeah, I'm glad you mentioned
that.
The
other thing about that bite,
I was just going to say, I'm glad you mentioned that, because I give a lot of presentations in all kinds of forums. And, that's always one of the questions I get. I'm sure you get it every time you give a presentation to the public as well. Is it true that baby rattlesnakes are more dangerous than adults? And, I've changed my answer to that over the years. And now I basically give the same answer that you gave, except I did not realize the component you talked about, that the babies actually, drop for drop, the venom is more toxic. And so I'm going to go back and find some of that work you were talking about. You said it was from your master's. Is that right?
Yeah, that was actually one of the early papers that I did in 1988, looking at venom ontogeny. So my master's work was primarily looking at Southern Pacific and Northern Pacific rattlesnakes and how their venom is when they're first born and how it changes as they mature into a large adult. Gotcha. And biochemically, it changes quite a bit.
Yeah.
And so one of those features is toxicity. It becomes less toxic.
Yeah, and we've had Dr. Young on the podcast talking about kind of the whole metering, question and all of that. So I'm glad you mentioned that. I will go look that up. But I'm really, really glad you mentioned that. And we're going to talk about venom, and we're going to talk about things like that, like, does venom change as snakes, turn into adults? Is that possible? These types of things. But anyway, so you're a high school student. You're have this dream job for somebody who's interested in snakes, and you're handling all kinds of venomous snakes, and you have this potentially traumatic, maybe exciting, I don't know, at least created a good story experience of getting bit by a Southern Pacific rattlesnake. And so I want to ask you, you're in high school doing this. Did you realize that as you going into college that this was something you could potentially do as a career? But before you hit that, you were about to say something about that snake bite, and I cut you off rudely. So please talk a little more about that snake bite, and then curious if you realized you could make a career out of this or not.
Sure. One thing, any kind of venomous snake bite on a good day is a bad thing, right? And so, no, the whole experience was very unpleasant unless you enjoy the sensation of hammers being pounded on your arm and throbbing pain and stuff like that, which I don't. What I was going to mention about the bite, though, is it was long enough ago that I did it back in the day when it was cheap. We didn't have insurance. My dad was in between jobs at the time, so I paid for it with my paper route money. And the whole thing, that is 6 days in the hospital, doctor bill, and antivenom, the whole thing cost $1,000.
Wow.
Now, even normalized for inflation, that was dirt cheap relative to what costs are these days. And so, people that are doing this free handling, not only is it painful, but you can incur a cost that can run $250,000 to $500,000 easily. And I've heard that from several different sources over the last 5 or 10 years. So it's an expensive accident. And so that taught me to not do dumb things that I was doing as a teenager. And strangely enough, teenagers occasionally do things that are probably not the brightest thing in retrospect. But as far as developing my career goals, actually in high school, I was very interested in language and took French, German, and Spanish and thought about becoming a UN translator. But when I applied to school, I declared a biology major because I was always interested in biology. And when I started at University of California, Santa Barbara as a freshman, that was the track I was in, and basically stayed in that track of biology, zoology, biochemistry for my entire academic career, or student academic career.
Gotcha. And so where did you end up? You've already said your master's was looking at kind of changes in venom in these two Pacific species and over basically young and old, comparing younger and older snakes. Does venom composition change? So where was that? Was that in California? Were you at a California-based university?
Yeah, it was kind of what's referred to as the Central Coast. So primarily it did include some sampling all the way down into LA County, Los Angeles County. and then through Santa Barbara and San Luis Obispo counties. So kind of that where California does the big bend and change over from a influenced by southern currents and northern currents at Point Conception there. That kind of area, in the greater Santa Barbara region and extending north and south. So that was where most of my sampling was from. And, we did some other species kind of on the side, sidewinders and red diamond rattlesnakes and things like that during my master's. But undergraduate and master's at the University of California, Santa Barbara. And then I started a PhD in what was called the zoology department at that time at Washington State University. And I had begun to look at the venoms in much more detail. And so the PhD was looking at the venoms in a lot more detail, taking them apart, describing the biochemistry and such, but also going very extensively into the venom gland structure and asking questions about how do they store this venom right next to their brain in this little sac of sorts for years without intoxicating themselves and having damage or having their venom fall apart. So that led into a whole host of other studies as well.
Gotcha. And where were you for the PhD?
I was at Washington State University.
You said that.
Which
I'm
was
sorry.
up in Pullman, Washington.
That's right. You said that. Sorry.
Yeah.
Great.
And it was a very significant change from Santa Barbara.
I bet. So, I've been up to Pullman.
In every respect.
Yeah, climate and just, everything. Yeah. So.
Yeah, it was a good school, but it allowed me to focus and stay in the lab, which I needed to do to finish.
Yeah, there you go. So you finished the PhD. Did you end up going directly to Northern Colorado, or were there any kind of steps in between there?
No, at the time, my wife and I moved to Southern Colorado. I got a job at Colorado State University as a postdoctoral research associate in the biochemistry lab of Anthony Tu, who was a world-famous venom researcher. He started working in the 60s and continued publishing all through the early 2000s. And he's retired now. He's still alive. I think he's either 93 or 94 living in California. But
great.
So I was working in his lab ostensibly to pick up more techniques in biochemistry. So
excellent. And then on to Northern Colorado.
Yeah, at that point, when I finished up there, I started teaching part-time at UNC, University of Northern Colorado in Greeley. And in 1994, started a permanent tenure track position, and I've been here ever since that time.
Yeah, gotcha. Well, let's transition a little bit, and let's start talking about what has been the kind of the center of your research program, and I'm sure a component of your teaching and all of that over time, and that has to do with venom. And so let's just start right at the beginning. And if I was to ask you the question, what is venom? How would you describe it?
Well, it's an interesting biologically synthesized material. And we mentioned a little bit about how the snake has to store it safely. We can get into that later. But it's really a mixture of different biochemicals, primarily proteins and peptides. And these are macromolecules, larger molecules that are comprised of strings of amino acids. And so when we talk about neurotoxins from cobras, for example, I say they're as natural as milk. Because milk has protein in it too. And those proteins just are strings of amino acids in different sort of arrangements, and of course are non-toxic. But we don't typically inject milk, so we won't go there. But it turns out that snakes haven't evolved this magical or de novo new stuff just out of thin air, but instead, if you kind of speak teleologically, snakes have looked around the body of vertebrates and said, I'll take some of those regulatory compounds, those regulators of all these different processes. And then in the venom glands, they overexpress those. So instead of having a regulator that's present locally, say in your bloodstream, and causes blood vessels to dilate, to expand, or to contract, instead of having a tiny, tiny bit there that would be used as a regulator, they give tens of thousands of times more. The net effect of a rattlesnake and other venomous snake bite is system failure. All sorts of different systems are affected almost simultaneously. And when we have a human that intercepts that bite, we get a very, very complex and very evolving sort of dangerous scenario that can be life-threatening and certainly is debilitating.
How about, so, and correct me if I'm wrong here, and I probably am wrong, but most or all of the venomous snakes are more recently evolved snakes. And so I'm just curious what you can tell us, what we know about the evolution of venoms, maybe, across taxonomic groups, but then more specifically with snakes. Is it indeed a more recent phenomenon, or is it something that we see going back, 80 million, 100 million years?
The answer to your question is yes and no, which is always satisfying. If you look at the origins of venomous reptiles, they certainly have a history of at least 100 million years. If you remember the Jurassic Park, that documentary on reptiles, I'm being facetious, they had a venom-spitting Dilophosaurus. That wasn't quite as far-fetched as it might seem. It did have tooth structure and such that suggests that it may have actually been a venomous creature. Whether it spat in the quantities that were in the film is debatable. But in fact, the basics are that venoms evolved in reptiles fairly early on. It turns out there's a couple of venomous mammals as well. So some weird things called solenodons, some of the shrews. Platypus males have a venom-conducting spine on the hind leg. And so we see venoms in a lot of different vertebrate groups. Well, in several different vertebrate groups, I should say. But really, the reptiles and snakes in particular have diversified that very extensively. If you look at the fossil record and ask when do we see vipers and cobras and things like that, there's good evidence from deposits that are around 25 million years old that these groups, we have good cobra-like animals and viper-like animals by that period of time. And their history is probably much, much more ancient than that. So venomous snakes have been around in one form or another for 75 to 100 million years. They have diversified into representatives of the families that we see a little bit more recently. But again, they have a fairly long evolutionary history. And that has allowed for these venoms then to have the geologic time frame necessary to diversify in all these different types of compounds. Okay. So when we say proteins and peptides in the venom, we're not saying one protein and one peptide. But if we really want to divide it out into the minutia, a venom can have as many as 100 or more different protein peptide components. Some of them are very similar to one another. Others are vastly different from one another. But it's a complex mixture that, again, is produced biologically by the snake.
And in that complexity, then those peptides and other peptides and proteins can be rearranged in different ways and create diversity of venoms across species and within species. And so that's...
and that's the name of the game, too.
Yeah. So speaking of diversity, snakes, when the average person thinks of venom, a lot of people probably have heard and think of the terms hemotoxin and neurotoxin, which would be broad categories of venom types that various species venoms may get put into. So the first part of this question is, do you, being somebody who knows these venoms as well as anybody on the planet, do you subscribe to that? Do you categorize venoms, and then or do you just think everything's so diverse that it's just a spectrum, or do you? And then if you indeed do, what are some of the? I mentioned those two that people probably think of most, but what are I don't know, whatever the top 5, 6, the top categories of venoms that you would think about? Reptiles face many challenges, from habitat loss to climate change. As an example, turtles are the most threatened group of animals on the planet, with over 60% of species classified as endangered. The Orianne Society is working hard to secure a thriving future for these incredible animals. To engage in the critical work we're doing, explore our website at www.orianne.org. There you'll find in-depth insights into the science, conservation action, and communication initiatives we're undertaking. Visit us today.
Well, when people use the term hemotoxin and neurotoxin, and they say something like a rattlesnake is a hemotoxin or a cobra is a neurotoxin, that's really a misnomer, because any rattlesnake species has all these different components. Some of them may affect tissues, some of them may affect the blood directly. Some of them, like in Mojave rattlesnakes, midget-faded rattlesnakes, tiger rattlesnakes, some of them actually are presynaptic neurotoxins. So they actually bind to channels and receptors on nerves and incapacitate the ability of the nerve to tell a muscle to contract. So when you say it's this or that, that really oversimplifies and is actually inappropriate for describing the venom. Now, if you talk about the clinical signs of envenomation, if a cobra bites you, it depends on the cobra, but let's just say an Indian cobra bites you, one of the major things you need to be concerned about right away are the neurotoxins that are in those venoms. And these are molecules called three-finger toxins because they look like three fingers pointing down. And they're very, very potent inhibitors of the receptor that's on our muscles that receives the signal from nerves that says contract. And so if you block those, then we have a situation of flaccid paralysis where the person can't move. And those molecules in the venom are small, so they move fairly quickly through the system. And they can be life-threatening because the phrenic nerve diaphragm that controls your breathing muscles can be compromised. And so you get paralysis of the breathing. And without treatment or without artificial respiration, iron lung kind of therapy, a person could, in fact, die fairly quickly from that. If we go to the other end, the hemotoxin, there are a few things that are actually toxic to the blood, which is how that term translates. But what I prefer to use is something like tissue-damaging venoms, because what we see in rattlesnake bites in particular in the United States is that most species produce very, very severe damage, local damage to muscle. And you may have seen pictures online, and I'm sure people have, where it almost looks like if you were bitten in the hand, you put your hand over a fire and toasted it. And it's all blebs and blisters and necrotic lesions developing and things like that. And those are due to some of the digestive enzymes, the specialized proteins that are in fact causing local tissue damage that then spreads and without treatment could take your entire limb or kill you for that matter. So although we can talk about the venoms as hemotoxic and neurotoxic from a clinical perspective, that's what we need to deal with quickly, neurotoxic or tissue damaging effects. Venoms are much more complex than that. And we also get into a situation where as we get into different clades of venomous animals, in particular the so-called rear fang snakes that are largely harmless to humans, but occasionally have produced some weird effects. Those and some of the front fangs as well produce venom components that are very, very specific to certain types of organisms. So for example, the brown tree snake. Many people have been bitten by the brown tree snake in the control efforts on Guam to try to keep it from spreading to other islands. And they don't show severe effects. They may show local reactions. Well, it turns out they have three-finger toxins that structurally look almost identical to what we find in cobras and coral snakes and some of the mambas and things like that. But it's a different molecule, and it doesn't really affect mammals to any significant effect. But it's a lethal toxin to birds and lizards. And so that got us very interested in that whole line of research into what we've referred to as taxon-specific effects of venom proteins. Because on on the one hand, if you look at the crystal structure where we have the whole molecule in three-dimensional space modeled of a cobra toxin and a brown tree snake toxin, and you overlay them, they're almost a perfect match. But if you look at the individual amino acids, go back to that as natural as milk sort of thing, if you look at the amino acids, those are different in those two different snakes' venoms. And that has the effect of being specific for this or that type of prey. So,
wow, that's interesting. So same, at least superficial structure, but you have the brown tree snake that really wouldn't impact mammals much, but does impact the birds due to the details, if you will, of the amino acids, and then the opposite for the cobra. That's really fascinating. So how about these other classifications people might hear? I was going to say, like, give an example of like cytotoxins or others. How would you? Do you have other major categories of venoms? And even if that's not how scientists would typically think about it, but maybe it's more like you were talking about, kind of in the treatment of and the symptoms of a particular bite, or do you classify those all under tissue damaging or?
Well, it's interesting that you bring up the cytotoxins in particular, because some of the spitting cobras have some very nasty effects locally that are not neurotoxic. And some of those effects are actually by these same kind of three-finger toxins. But in this case, they're not binding to components of the neuromuscular system, but instead they're causing damage to local tissues, cytotoxic effects. Cytotoxin simply means cell toxic or cell damaging. And we've done a lot of work with cancer cells in the lab. We can grow them in the lab up to millions to tens of millions very readily in a couple of days. And then you can ask the question, what effect does this venom or that venom have on the viability of the cells? Does it kill them or not? And a lot of the different snake venoms, whether we're talking about the elapids like cobras and their allies, or we're talking about the vipers, rattlesnakes and such, kill cells very nicely. So they have cytotoxic effects, but the cytotoxic effects on those cells are different in some of the different venoms. It also turns out that some of the most dangerously toxic rattlesnake venoms, such as the South American rattlesnake, Crotalus, that is not particularly toxic to cancer cells at all in the lab. And you put a fair amount of that material on them, and it doesn't have much of a killing effect on the cells. If you put that into a human or into another mammal like a mouse, it's very rapidly lethal. So there's some differential effects that we can have in the lab, in vitro or in ex vivo, compared to what we see in a whole animal envenomation. So we have come up with a dichotomy in rattlesnake venoms that early on held very nicely type 1 versus type 2 venoms. Type 1 being more digestive function, type 2 being less in the way of the metalloproteinases that causes structural damage and a lot of hemorrhaging and things like that, but has instead neurotoxins in the venom, and these presynaptic neurotoxins. It turns out that there's a lot of gray zone in there. Things in biology never fall out as this or that. And you can set up those ends of a continuum, but there are a lot of things in the middle there. Mm-hmm. And as we've gone into more and more research in different species of rattlesnakes, patterns that we thought were generally applicable, like the venom ontogeny effects that we saw in the Pacific rattlesnakes, don't necessarily apply to other species of rattlesnakes, even fairly closely related species.
Yep. Well, you've talked about how, first of all, there's a diversity of venoms, and that in talking about how they impact whatever type of, say, blood cell or tissue cell, that you've talked a lot about receptors. And so I'm curious if you, in a very, imagine I'm a sixth grader, tell me how biochemically, and this is a generalization, and it may be so different across venoms that you can't describe it, but get to that level, the level of like you've got a venom molecule, and you've got a receptor on a cell. Like, how does venom biochemically destroy a human cell, or maybe destroy is the wrong word? How does it impact a human cell?
Well, again, what we have are a whole bunch of different molecules in the venom. And these are really modified regulators of all sorts of different processes in the body. And so these various molecules affect very different sorts of parts of the systems. And one of the reasons I don't like to say, well, a venom is cytotoxic is it can have that effect, but there can be many of those molecules in there that don't bind to cells at all. Instead, they bind to things like there are proteins that make up our blood vessels, right? Uh-huh. And that holds the whole thing together. Our blood vessels are comprised of cells, but they have specialized molecules, proteins that act to hold it together. And so some of these venom components, and these are referred to as metalloproteinases, are enzymes, and enzymes are proteins that catalyze reactions that otherwise wouldn't happen. And they can keep doing that over and over and over again. And so those venom metalloproteinases are attracted to that surrounding layer of protein that holds blood vessels together, and they begin to break it apart. They have particular parts of the molecule that act as, you can think of them as molecular scissors. And so they're cutting apart the structure. Well, the blood vessel itself is under pressure as well. And you start chopping apart the things that hold that tube together, and it begins to rupture. And when it ruptures, that produces what we call a hemorrhage. And that can affect all sorts of different systems, because now blood is not staying pressurized and staying in the system, but it's leaking out, blood pressure can fall. There are other components in the venom, same venom of rattlesnakes, that actually cut a little protein that's circulating in the blood. You cut a big protein, they cut a little peptide out of it. And that has the effect of causing our blood vessels to relax or dilate, become bigger. And if that happens suddenly, it can lead to a cataclysmic crash in blood pressure. And you may have heard about the Gila monster bite that occurred in the Denver area that killed a guy just this last spring. And in part, Gila monster venom has a large amount of that particular type of enzyme in there. Mm-hmm. He apparently got a very large dose of venom. And one of the first things that happened is that he just almost passed out, fell to the floor because his blood pressure crashed. And things went downhill from there. He had ended up dying from the bite. It was terrifically horrible.
Yeah, so these venoms, again, we're just speaking about a general snake. The venoms are very diverse, very complex, hard to categorize, as we've already talked about. And so then talking about kind of the mechanics of some of this, then this venom goes into a human body or whatever, a prey animal's body. And there's different components of the venom that act on different parts of the body. You might have some attaching to a blood cell. You might have some, as you said, attacking proteins around a vessel. You might have others that are going off and attaching to, some type of nerve or whatever it might be. That's interesting. And so there must be all of that biochemistry must happen at kind of different rates, if you will. And so there must be kind of a natural progression of symptoms to a snake bite, depending on, say, the species and maybe a lot of other factors. But is that something you see in snake bites where maybe you get some initial light paralysis, and this is completely hypothetical, right? And then you start to see tissue breakdown around the area of the bite, and then later on they'll start losing their sight or whatever it might be. But do you get this kind of natural progression with certain species where you have all these molecules in the body, they're affecting different things, and those happen at different times, or am I completely off, my rocker here?
No, I think that's a good generalization. Now, I have to state that I'm not a physician, and I've experienced snake bite, but I haven't treated it personally. But from discussions with a lot of clinicians that do emergency medicine and actually work with these things, yeah, this is a progressive issue. And Dr. Sean Bush, a number of years ago, came up with a little adage, time is tissue. And that's meant to be that indicate that what you should do is get to clinical help, get to a hospital as quickly as possible. And the longer you have in a delay of that time, the more of an effect is going to be unchecked. You don't have your own antivenom typically that you would inject. And so when a snake bites, most of the snakes will strike, envenomate, and release. And rattlesnakes can do that in about a quarter to a half of a second. So they're very, very fast. What they do is release a large bolus of venom that you can think of as is now deposited as a depot, typically under the skin. If for some reason it happens to get into a blood vessel, that can be very problematic very quickly. And we've had some incidences that seem like that's what's happened and had fatal outcome from something like a prairie rattlesnake a couple of years ago here. So if it's deposited under the skin, not in a vessel though, it's going to diffuse out from that site. And so the effects you're going to see locally are going to be most extreme. And then what we see are various aspects depending on the type of venom it is. And so if it has neurotoxic components in its venom, then we'll start seeing neuromuscular functioning compromised. And that can, in fact, be manifest in problems with blinking, with swallowing. And of course, if it goes systemic and gets into the body proper, then we start having breathing problems as well. And that's where we can have, if a cobra gives a good bite, delivers a lot of venom to a person, then those molecules diffuse through and they can get to the site of the phrenic nerve diaphragm and compromise breathing muscles fairly quickly. And if that happens, that can kill a person fairly quickly, half an hour to an hour. So yeah, the whole aspect though is that things tend to get worse before they get better. And when you go to a hospital and seek treatment for something like a rattlesnake bite, you have an expectation that, okay, now you're at the hospital, things are going to get better. But that venom has been in place for some period of time, ideally a very short period of time, perhaps hours or even longer. And so as that happens, not only does it have effects on the tissue that's closest, but it also liberates things from your own body and your own cells that can be promoting damage as well. And so again, that's why there is this adage of getting to a hospital as quickly as possible. And the idea of the best first aid are your car keys. Get in your car and get to the hospital as quickly as possible. Yep. Because that's really, antivenom therapy is still the major therapy that we have available.
Mm-hmm. A couple more questions about venom, and then I want to talk a little more specifically about some of the research you've been working on with venom. But we talked about diversity of venoms, and I think we have kind of been talking about it in a sense, kind of like, say, among taxonomic groups, among species or families even. But talk a little bit about venom diversity even within a species, both maybe space and time. And what I mean by that, I'll take animal I work with quite a bit, timber rattlesnake. So, I know there's been a fair amount of work looking at venom in different parts of the rattlesnake's range. So that's what I mean in space. Is there diversity? Does it vary? And then in time, maybe talking a little bit about, some of the types of things you've worked on, say, during your master's research.
Okay. Sure. Well, one of the things I've always kept in mind is you should always have something in your backyard to work on, right? Something that's convenient and proximal to where you are. And so we have 3 species of rattlesnakes in Colorado, and we've worked with all of them and venoms and aspects of their biology with all 3 species, but we've primarily been focused on Massasauga rattlesnakes, which are a species of special concern in the southeastern part of the state, and midget-faded rattlesnakes that we have a field project going on right now that's funded by BLM looking at distribution and relative abundance in the western part of the state, and primarily in the northwestern part of Colorado and then into Utah and the Canyon Country. But it turns out prairie rattlesnakes, which we kind of were ignoring because they're very widespread and common, these other things are sexier snakes to work with. Turns out that the patterns that we see in prairie rattlesnake venoms are really quite interesting. And one of my PhD students that finished up a couple of years ago, Kara Smith, who's now postdocing in a lab down in Anschutz Medical Center in Denver, was the one that spearheaded this work looking at prairie rattlesnake venom and its composition all the way from right at the edge of southern Canada down to the edge of Mexico. And it occurs very broadly across the Great Plains. Long story short, what she found is that right in the vicinity of Interstate 70, and we just use that as a marker because most people know where that interstate is, the venom shows a very disjunct change. And so north of that, the venom is dominated by low amounts of these tissue-damaging components, the metalloproteinases, but high amounts of a small peptide toxin called myotoxin A. South of that, we see the opposite situation, that these digestive enzymes, the metalloproteinases, are much, much higher, and the amount of that myotoxin A is very, very low relative to what we see north of that. So right in the middle of Colorado, we see this disjunction in the venom composition, and that northern phenotype or northern type of venom that we see is like that all the way up through Montana right into the border with Canada, and that southern phenotype that we see on the south side is like that all the way down to the Mexican border. So that begs the question of why this big difference in venom composition. And Kara was very good at trying to get at that, looked at differential prey use and things like that. Long story short, again, we're not completely sure what the driving force is for that kind of difference, except perhaps during glaciation events, a lot of the ranges of rattlesnakes were shifted far south. And then after the glaciers retreated, snakes moved north. And we see that in a lot of the snakes as well. So it's possible that associated with that response to changing climate, there was also a change in the venom composition. But that's pretty hypothetical. There's some kind of loose correlations with difference in prey use. So in the northern groups, we see almost exclusive use of mammals. In southern groups, we see mammals, but also lizards are eaten. Lizards are much more abundant as you move farther south as well. Different species are found in those areas. So there is also a possibility of one of the factors being this more diverse prey, leading to a different type of venom in the southern snakes. So there are a number of examples like that as we go around the world and look at species that show this north-south latitudinal difference in distribution. India has cobras in the north and the south. Their venoms are different enough that sometimes the antivenom that's produced mostly against the southern is less effective against the northern, and some of the east-west differences as well. So it has practical consideration. So the prairie rattlesnake turns out, we're kind of looking down our nose at it initially, but it actually is a very interesting species to look at in more detail.
You mentioned as part of that description of the prairie rattlesnake venom, you mentioned myotoxin. What is a myotoxin?
Mm-hmm. Well, again, there's debate on where exactly that molecule hits, but it's not an enzyme. It doesn't catalyze reactions, doesn't chop up proteins and things like that. Instead, what it seems to do is bind to a particular type of protein on a particular part of the muscle that produces the effects. And this is based on work when I was a postdoc with Anthony Tu at Colorado State University. He had a PhD student, Ponsak Utaisincharon from Thailand, who did some really good work looking for where does that molecule actually affect skeletal muscle. Because what we see is that mice that are either injected with that or are bitten by prairie rattlesnakes in the northern area, one of the first things that happens is they hyperextend their limbs and they show what's called a tetanic paralysis. So their muscles are contracted, but they can't move at all. And it happens, it immobilizes mice just like that. So a prairie rattlesnake from our area of Weld County, Colorado, if it hits a mouse, that mouse isn't killed immediately, but is stopped almost instantaneously. So it's a very potent toxin from that perspective as far as stopping prey, which is what snakes want to do, right?
Stopping mammals or stopping all prey?
Well, that's another interesting aspect of the molecule. We've done comparative toxicity studies, both in animals, but also in tissue baths, and have shown that it has almost no effect against lizards. So you can give a very high dose to a lizard like a fence lizard or earless lizard, has no effect on it. Lizards are primarily eaten by smaller snakes. It's just a size-based thing. Adults will take things opportunistically, but they typically want to feed on mammals, bigger package, better protein load and all that kind of thing. Well, it turns out that in the baby rattlesnakes in the same area where the high myotoxin levels are in the adults, they have half or a third as much of that myotoxin as the adults. And they actually show more in the way of those digestive enzymes in their venom than the adults do. So that pattern is kind of backwards from what we saw in the Pacific rattlesnakes when I did my master's so many years ago. So
yeah,
it's a rather interesting thing. There is this association with dominant prey type, venom composition in some cases, but yeah.
If you enjoy listening to the Snake Talk podcast, stay in the loop with the Orianne Society's latest conservation updates from the field. Read about new published research, as well as fascinating insights on the species we work with by following us on social media. Follow, like, share. Let's spread the word. Together, we can make a difference. Find the Orianne Society on Instagram, Facebook, X, or LinkedIn and be part of the conversation. Well, let's move on and talk a little bit about kind of some of the work you've done in your lab relative to, you talked about practicality, like the uses of venom, whether that be for, say in the medical field, human health, or other kind of uses. And so maybe I'll just kind of leave it at that. But maybe you could start off by talking about venom. We've talked about how complex it is, but why has venom been a fruitful place to examine for all kinds of practical uses, and then maybe kind of get into some of the things that you've been working on over the years in that arena.
Well, interest in venoms and venom actions and composition and what's in all these venoms has been of interest to people for a very long period of time, primarily because when they get bitten by venomous snakes, terrible things tend to happen. And before we had modern medicine interventions, there wasn't a whole lot that people could do. Chop off a limb, hope for the best, those kind of things, come up with some kind of magic incantations or something. But as we move more into the modern world of analytical chemistry, biochemistry, molecular biology, and then into the genomics, proteomics era, our ability to go in and ask, what is that venom made up of, has increased tremendously. And as that has happened, it turns out that venoms that have 100 different components, for example, those are not 100 different proteins. They're only about 15 or 20 different proteins, but the genes that code for them have been duplicated, in some cases many, many times. And so we have all of these different genetic copies that are slightly different from one another. Well, this is exactly what a combinatorial chemist does if they're looking for drugs for treating whatever malady we might think of. They're looking for things that are closely related. They typically have some idea of a compound that might go in and interact with a cancer cell, let's say, on a surface-based protein in a certain fashion, but it doesn't really do what they want. And so changing the chemistry of that molecule slightly, trying the experiment again, again and again. Well, snakes have done that over the last 50 to 100 million years. They've tinkered around evolutionarily with these molecules that hit all these different systems. And so, again, we have one class of proteins, say those metalloproteinases. And one of the things that interested me very early on was, okay, if you want to use something like that to digest blood vessels and tissues of prey, that's great. Why not produce a huge amount of one? Why are there 3 different classes of them and perhaps 15 different versions of all those in a single venom from a single individual? And in part, it's because there's this dynamic play between predator and prey. So if the snake's on top, the venom works really well, kills the prey quickly, it gets a meal, the snake's happy, the mouse is not. But that sets up a selection scenario where if there is something in the physiology of the prey that gives it greater resistance to that venom, those mice are going to be survivors. Okay? So we get this dynamic back and forth complex interaction between predator and prey that some people have referred to as this coevolutionary adjustment back and forth. And that has, in fact, largely been responsible, we believe, to why there are so many different venom components. Going back to using those as therapeutics or possible drugs, again, those various variants have already been provided for us. Now, back in the 60s, I came across a paper where cobra venom, crude cobra venom, was being used to treat schizophrenia. At that time, they knew that there were neurotoxins in the venom. Schizophrenia had to do with the nervous system, so maybe put those together and something happens. Well, it's a very simplistic sort of mindset. And the analogy that I use is you can remove a tumor from someone with a shotgun, but that's going to cause a lot of collateral damage. And that's how these venoms are. On the other hand, if you purify those venom components, characterize them carefully, and have some idea of what you're looking for in all those different variants that have evolved in a prairie rattlesnake or a timber rattlesnake or your favorite venomous snake from around the world, those may actually have some use in doing something very different from what the purpose was that evolved. And I can give you an example of that. That involves molecules that are referred to as disintegrants. So a number of years ago, in collaboration with David Chazar, who is a professor of psychology down at the University of Colorado Boulder, he's since passed, he and one of his students, Anthony Saviola, and I worked on this project together, asking the question, okay, David showed that snakes, once they've envenomated a mouse, can actually follow the envenomated trail preferentially and go after a mouse. And if you do Y mazes and all these other types of behavioral tests, you can show that very nicely. The basic question was, how do they do that? And so what we did, we began taking the venom apart. And we hypothesized that some of these proteins that are toxic and cause all sorts of horrible tissue damage and changes to prey would be the ones responsible for that. Turns out that's not the case. There's a little molecule in a small amount called a disintegrant. It's not even very toxic. If you put it in with cells, it doesn't really kill them. That molecule by itself, when injected into a mouse, allows the snake to be able to relocate a prey that was envenomated, even though it was just a fraction of the venom, not the entire venom. So the snake has this relocator protein in its venom, which makes sense. And we only find it in vipers. We don't find it in snakes that are strike and hold feeders like most of the elapids. People have looked at disintegrants, and Anthony decided to look at disintegrants in more detail as well. It turns out that they have some very interesting properties for treating cancers. And in some of the cancers, what happens is when the cells begin to migrate away from the site of an initial tumor formation, they become metastatic. They actually have little legs called integrants that they use to move out. And they digest their way through tissues and such to move about the body. And that's when cancers become very critically hazardous. Well, these disintegrants actually bind to those integrants and they inhibit the ability of those cells to move around. And Anthony showed that with a molecule from a tropical rattlesnake that we got on a confiscation years ago, and did some very interesting work in the lab in working with cancer cells, and showed that that molecule by itself inhibited metastatic activity or movement activity of cancer cells. So here we've got something that has potential as a therapeutic development, and there's a lab in USC in Southern California. Frank Markland, he did a lot of work with one from copperhead venom. And in fact, here it has anti-cancer properties perhaps, but the snake didn't evolve this. This didn't evolve in the snake for our use for treating cancers, right?
Yeah.
It uses it for something very, very different. So it's kind of an interesting example of where basic biology, basic chemistry can inform applied chemistry. Okay? So we've got these two things that are seemingly different, but involve the same molecule. So.
Yeah, that's fascinating. So any other examples that you've been involved with of say, snake venom being developed, whether it's in the medical field or something else? I mean, I know there's a lot of examples of, I've heard like mamba venom being used to develop medicines for ALS, or Gila monster venom, or whatever it might be, blood thinners. But are there any other examples that you've been involved with that you think would be worth kind of mentioning?
Well, we're really in the business on that end of things of evaluating molecules for drug discovery rather than clinical applications. And this is where when some of our work has gotten into the lay press, newspapers in particular, we get calls from people asking if they can enroll in a therapeutic trial or something, and we don't do that at all. I mean, that's a whole different ballgame. So we have worked with a number of different venom proteolytic enzymes, these ones that clip proteins, cut up proteins. Amongst them, we talked about the metalloproteinases. Those kind of wreak havoc with blood vessels and tissue and things like that. But there's another group of them called the serine proteinases, a different type of protein that tends to be much more specific in its action. And again, we've characterized a number of these from things like midget-faded rattlesnakes and from Russell's viper from India. And I had a visiting scientist here for a year that did a lot of work with one from the Russell's viper in particular. And that has the possibility of being developed as a anti-clotting agent, if you will. And that has certain applications for times during surgery, et cetera. You might want to drop one's ability to form clots, but you don't want to keep it depressed, but just for a short period of time. So we've had a little bit of that. It's more tangential, not really direct therapeutic development, but some ideas here along that line.
Great.
You mentioned the Gila monster, and there are two blockbuster drugs that have been developed from Gila monster venom. One of these is Byetta, that's been used to treat type 2 diabetes, and then more recently, a very similar molecule, Ozempic, that also was developed for type 2 diabetes, but is used for obesity control. And in that role, that thing has gone off the charts as far as sales and such. So there are some compounds that are out there that have been developed from venoms and are very, very successful drugs. There's an ACE inhibitor, captopril, that was developed based on some peptides found in South American vipers, venoms. And that probably is the single most important drug from venoms as far as how many people globally are helped with high blood pressure from either that drug, captopril, or some of the spin-off secondary drugs that have been developed based on that. And then, again, that comes back to the whole venom. The whole venom is very, very damaging because it's kind of like that shotgun blast. But here, if we look at particular molecules and we know a little bit about what to look for, we can actually find some things that have very good therapeutic value. So.
Well, we've been going almost an hour, so I don't think we're going to be able to get into the topic of kind of genetics, ecology, natural history, but just wanted to recognize that you do also work in those arenas. But one last Venom question, then we're going to begin to wrap this up. So we've talked a lot about Venom. We've talked about different research projects, both, whether it's more towards evolution and or towards very practical applied uses. What do you see as, I'm assuming you'll be doing research for a good number of years now, but you're on the other side of the curve in your career. What's the future of Venom research? What are the big things that we need to know or people are starting to look at? And that could be just in the basic biology of venoms all the way down to some of these practical uses.
Well, it's interesting. As we develop more sophisticated analytical tools, we can ask the same questions and probe that much deeper, right? And so as proteomics and genomics have become much more commonplace, we can ask questions like, what's the venom made of? What are the genes that code for it? How are those regulated, et cetera? And they can also, snakes and snake venom glands can be used as models for all sorts of other types of processes, secretion of materials, et cetera, things like that would occur in non-venom systems as well. As far as evolutionary questions, wide open, what are the driving forces for, first, evolution of venom? We have some ideas on that. Being able to stop something chemically by injecting it and getting out of the way while it's going through fits until it's mobilized. Direct advantage there. Why do we see all these different types of venoms? If you look at a cobra venom versus rattlesnake venom, the components are really quite different. There's some shared things, but there are a lot of things that are unique to each one. Why do we see diversification along that line in that lineage and along this line in a very different lineage of snakes? So all sorts of different evolutionary questions we can start addressing those in a lot more detail now. More recently, there's been a lot of interest in small molecule therapies for treating snakebite or alternative therapies to antivenoms. And we've had an opportunity to interact with a number of different groups, and this is where collaboration between people that have different skill sets has led to some really cool and really rapid advances in our understanding. And so we're seeing molecules that were used in clinical trials for treating cancers or treating some other disease. They failed in clinical trials, but they actually are targeting molecules in cancers that are very similar to what we find in venoms. And so can we repurpose those drugs that have already gone through some clinical trials in humans as far as safety and apply them to a different situation and system? And that's what's happening right now. Even more interesting is the use of what are called mini binder proteins. So there's a group in Seattle at the University of Washington that are essentially wizards at doing in silico design. So instead of having wet bench chemistry trying this and trying that of all these different variants of molecules, they're able to model those, the toxic molecule, and then something that might neutralize that. And we've collaborated with that group. We have a paper that's under review in Nature right now that hopefully will be published shortly that shows these mini binders are incredibly potent inhibitors of some of the most hazardous three-finger toxins, these neurotoxins that paralyze and kill animals and including humans very, very quickly. And they're useful up to a half an hour after, we've only gone to a half an hour, but they're useful as a therapeutic a half an hour after a toxin has been administered. And so that's really encouraging as far as here's something that may not replace antivenoms, but in fact can be used as a first aid therapy, can be given as a IM shot in your arm, just like you'd get with a vaccine or something like that. It's a fairly stable molecule. It doesn't require refrigerators. So we can, that problem of having a long period of time between when the bite occurred and when you reach treatment, we might be able to shorten that in rural areas around the globe where snakebite is most problematic to a much better chance of surviving the bite and also decreasing morbidity. Because you can survive something like a soft-scaled viper or puff adder bite, but you may lose your limb. And if you are an agrarian individual that greatly compromises your livelihood. So there are a lot of different areas like that in snake venom biology. A lot of people have gotten interested in the field in general because there's a nice correlation between the phenotype, the genotype, and the fitness component. If a venom doesn't work, it's not going to be too useful for a snake. So,
well, I like, as you know, I like all of my guests to tell me a good snake story. So, what's your? Do you have a good snake story for us today, Steve?
A good snake story. Well, I guess there could be a couple of them. But one of the most profound ones came from my earlier childhood, that is my teen years, working at this company that supplied zoos and schools. And as I mentioned, took care of all sorts of venomous snakes. One set of these were 3 king cobras that we had in one cage. The small ones were 11 and 12 feet. The big one was 14 feet. And it was my job to clean the cage. And so I moved these from the cage to a trash can. One of the snakes, the biggest one, 14er, would have nothing of it. And so for the next 20 minutes or so, we did this pirouette dance of sorts where the snake would hood about 4 feet off the ground, chase after me. I would get outside the door, close the door. The snake would turn around. We'd go through the same thing, trying to get it back under control. So that went on for, like I say, 10 or 20 minutes. One time, I didn't get the door closed quick enough, got out of the door, got out of the secondary door into the main room where we have the harmless snakes. And we were able to corral it into another room that was for amphibians that was colder. That's when I decided, well, let's see, $3 an hour. This is probably above my pay grade right now. Called the boss in. He ended up pinning it down and we contained the snake. So no one was damaged. No snakes were injured in this whole adventure. But it was a fairly profound demonstration that, some of these snakes have a lot more going for them than we might give them credit.
Great story. Now, hearing that story and also, a lot of what we talked about, just thinking you must be like an Ayacook's nightmare in terms of, you're subjecting certain animals, you're injecting them with venom, and then, the human safety component. For those listeners that don't know, an Ayacook is like a university entity that looks at animal welfare issues and human safety and all of these things. So I'm sure you deal with that quite a bit. Maybe you probably chair the committee. Just
oh, yeah.
So anyways.
I've been on the committee and I'm an advisor to it. I've been here for about 30 years, and in that time we've handled thousands of snakes and we've had no accidents. And so, having an unblemished record of working with venomous snakes does help considerably.
Yep.
So
yeah, that's great. And you've only had the one bite, it sounds like, or you didn't tell any others. So yourself,
yes.
And I've never had a venomous snake bite, and mostly have handled vipers, but I've had bushmasters and some other more serious vipers over the years. And but I've probably had, trying to calculate it, probably over 10,000 handling events. Some of those are same snakes over and over, and I've never had a bite.
Yeah.
And to me, that's a badge of honor. People often, it's always a question: did you get a snake bite? Or, people want to talk about that. But the idea of not getting a snake bite, that's what I'm proud of. Not, I don't want to show off the rattlesnake bite I got. So they make for good stories, but I'm glad I've never had it.
Yeah. When Anthony gave his defense seminar, a question at the end was, have you ever been bitten? And he said, no, and I intend to keep it that way. And the person was like, well, doesn't that give you some street cred if you've been bitten? And he was like, no, it does not. It means you were careless.
Yeah, there you go.
And so we try to keep that in mind. And again, snakebite is something you should experience vicariously. Go online and see the pictures and let it go at that. It's really, on a good day, it's a miserable experience. On a bad day, it can leave you permanently damaged or dead. So free handlers, no reason for doing that.
Well, Steve, thank you so much for joining us today on Snake Talk.
My pleasure.
And I just wanted to thank the audience and tell everybody to remember snakes are animals too. And it's a privilege to see one in the wild.